Hello. This is SciFi Talks, and today we have our special guest, Dr. Sanjivan Gautam. Dr. Gautam is an Associate Director of Division of Immuno-oncology at Astrazeneca USA. He is also a co-founder of Switch Therapeutics, Germany, and NCI and Nepal. Dr. Gautam has long experience working on T-cell biology in both academia and industrial settings in India, Germany, USA, and prestigious organizations like NCI and Gilead. We would like to proudly welcome our very talented, always inspiring guest, Dr. Sanjivan Gautam.
Welcome, sir.
Thank you, Sudarshan, for the invitation. Wonderful to see you again.
So how do you define immunotherapy, sir, in very easy language, in a layman term? So how do you define immunotherapy?
Right. So I think the word itself tells you what does it mean. So it's immuno and therapy, meaning like if you use any component of your immune system, your defense system, internal defense system to treat any sort of disease, right? That's called immunotherapy. For example, if we talk about a very popular immunotherapy modality these days, the checkpoint inhibitor molecules, right? Pembrolizumab, nivolumab, durva, right? So these are all antibody molecules. So what they do is, they go to the tumor microenvironment and they inhibit the function of inhibitory receptor on the T cells, right? So what we are doing here is we are using the antibody molecule. So antibody molecule is a part of immune system, so that's why it's immunotherapy. Now also, if we talk about cell therapy, so there are different types of cell therapy, and if we talk about, let's say, tumor infiltrating lymphocyte therapy, so what we do here is we take out T cells from the tumor that are reactive to the tumors, and we expand them in lab and we infuse back into the patient. So here also we are using our own immune system, which is tumor infiltrating lymphocytes. So it is also part of immunotherapy. So it's also another form of immunotherapy. Likewise, there are other forms of immunotherapy as well, where we can use the cytokines, where we can use the vaccines and things like that. So overall, immunotherapy is a type of therapy where you use your immune system to treat any sort of indications or disease.
What do you think, how did this immunotherapy came into rise? Now, the people are talking a lot about the immunotherapy, not just in the cancer, but in the different kind of disorders. So what is the main reason behind this, behind the rise of the immunotherapy?
Right. I think this is a very good question. So, everyone asks me this question, why you think it's rising and why it's so popular these days. And one of the reasons here is because, we have seen the, very good response by the patient who are terminally ill in different indications like cancer, right? So this thing we have seen after the immunotherapy. So what I mean to say is, back in 2020, 2000 or 2002 at NIH, there were some patients treated with cells, immune cells, and these patients responded very well towards those therapy, right? And that was very encouraging for everyone in the field. They thought like, yeah, maybe, this is the path forward to treat many indications, including cancer, right? And when it started from there, and many people started to explore other aspects of immunotherapy, for example, checkpoint inhibitor, and we know this modality has already been given a Nobel Prize back in 2018, right? It is because there was a very good response rate seen in patients that were unresponsive to existing therapy. And the another example is a very popular example is the chimeric antigen receptor therapy. So back in 2010 or 9, the pediatric patient, Emily Whitehead, when she received this therapy for the first time, she showed a very good response, and then, her disease was eliminated. She had a B-cell cancer. So due to all these kind of observations, which were in exploratory phase, people got so much of encouraged, they got the encouragement, and that's why I think they started to think about, yeah, maybe this is the path forward to treat cancer. And then, that's how it started. So people started to explore the different aspects of T cells, NK cells, macrophages, and they, try to understand how the function of these cells can be exploited to treat diseases, right? There are different types of immune system, there are different types of diseases, different types of kind of antibody molecules and, cells. So everyone is exploring that only because, some of the studies in the past had shown a great promise in these modalities. So that's why I think like it was based on the evidence. Yeah. These therapies can cure the disease. It not only treats the disease, it has the curative potential. Okay. And that was the most kind of a promising factor for any drug developer or any scientist, right? And on top of that, here the interesting thing is in this field, if you talk from the scientific standpoint, you have the ability to explore or do science And also you have the ability to bring this understanding of your science directly to the clinic. And that could be another factor why the scientists who were doing basic immunology, basic cancer biology, they are switching towards the immunotherapy. It has the practical implication. And it's always fun to see when your science works in the bench as well as in the bedside, right? So that could be another, professional reason why people are switching towards this therapy and it's getting more popular. And the another point could be, mostly the immunotherapy, you will be using, the human's own product, right? So you are using, like, let's say in case of CAR T cell, autologous CAR T cell therapy, you are taking out CAR T cells from patient, and then you are genetically engineering these T cells. You grow these cells in the lab and you infuse back into the patient. So meaning, like, the patient is taking its own product. So that's why there is a very less chance of getting higher level of complication. So that could be another reason why people think that it is a promising modality. Yeah.
Okay, sir. And what kind of research do you think is contributing to the success of immunotherapy? Like antibodies, like we are very far from the antibody research. I think we have done a lot in the antibody research. Do you think this antibody research and like engineering principles in the biology is a contributing factor for the success of immunotherapy?
Yes. I think it depends on what kind of modality you are talking about, but certainly I think the development of antibody technology has contributed a lot in this field. And if we want to talk about like what are the major fields that have contributed to the success of immunotherapy, I would like to mention three different areas. The first one, as you said, is the synthetic biology or the antibody technology, right? In the industry setting, we call it antibody campaign.
Okay.
So this is one of the major contributor because the antigen recognizing property of immunotherapy is achieved through antibody molecule, right? That's the first thing. And how you design this antibody molecule, how you manipulate the SCFV region or how you manipulate the hinge region, transmembrane region, these are very crucial factor in the success of immunotherapy. So certainly the synthetic biology is very, very important here, especially for the CAR T cells, for engagers, and also for the TCR T cell field, right? Now, the second one I would say is the immunology itself, the basic immunology. Once you find out the right synthetic molecule for your drug product, you want to use the right cell product as well, right? So the basic understanding of immunology is very important here. We know there are so many cells that exist, T cells, NK cells, macrophages, but what is the right population that gives you the optimal outcomes? That is also very important, and that understanding comes from the basic immunology. So that's the second one. And the third one is the genetic engineering. Right. So now what kind of antibody molecule is useful for you, and you now know what kind of cellular platform you are going to use. Now you need a technology to insert that synthetic molecule into your cells, right? So the advancement of genetic engineering technology, for example, lentiviral particle, adenoviral particle, tail nucleases, Sleeping Beauty technology, CRISPR technology, these are also equally important. So I would say these three different topics are the major contributor to the success of immunotherapy and cell therapy.
So when we talk about the cellular immunotherapy, the CAR T immunotherapy comes first, like the T cells, mainly based immunotherapy based on the T cells. Why do you think the T cells is the most favorite kind of therapy rather than macrophage? Yeah. Is it because we are not looking at other therapies, or why do you think? Is it by the coincidence, or why do you think?
I think that's also a beautiful question. So, being a T cell biologist, what I feel is, of course, T cells are the most beautiful immune cells in the body. However, to answer your question, we need to talk about the facts as well, right? Yeah. So, I would like to start from the source, right? So, if you take a 100 milliliter of the peripheral blood from the patient or from the donor, you can get up to millions and millions of your T cells out of that 100 milliliter of blood, which is enough for you to generate the CAR T cells, right? And also, these T cells are very easy to activate in vitro in the laboratory.
Yeah.
And also, these are non-adherent cells, so they grow very well in the culture plate, and also it's relatively much easier to grow in the system like Protege and Cocoon, which are the CAR T manufacturing platform or TCR-T manufacturing platform. And also, they expand a lot in vitro so that you can have enough cells for the therapy. So overall, to answer your question in short, T cells are very easy to handle, very easy to genetically manipulate, and very easy to expand, and very easy to express the synthetic molecule. That's the reason why it's a very lucrative cells for any sort of cell-based therapy. And also it has a very good ability to kill the target cells, especially the CD8 T cells. CD4 T cells also, they have the ability to kill, but their major function is the release of interleukin 2 that helps the CD8 T cells to sustain and function better in the tumor microenvironment. So that's why T cells are really the best cells for this therapy. Having said that, there are also other cells that are being explored, like NK cells, right? So NK cells, one of the advantages of NK cells over T cell is, you don't see much graft versus host disease if you infuse these cells into the patient, even in the allogenic fashion, right? But the trouble with these cells are, they are not easy to manipulate genetically, and they don't expand much. And also, there are very few evidence that it works in the clinic. There's only one clinical study that came out from Baylor College of Medicine in Texas, where they have used CAR NK cells to treat multiple myeloma patient. And also other cells like macrophages are also in use, but I doubt that it will be as efficient as T cells because they don't expand much, and then it's a very plastic cells, right? They can convert to M1 and M2. If it is M2, then it will be a suppressive phenotype. So instead of giving you a therapeutic benefit, it may, give you a negative consequences there. So it is still a kind of a, very preliminary phase, and we are looking for more data to understand the function of these cells in the context of immunotherapy. So due to all this reason, I think why T cells are the most favored cells for this sort of therapies.
I'm slowly moving towards the solid tumors complexity, but before that, I would like to talk about the TILs. I think it was FDA approved very recently, the TIL therapy. So do you think we should move towards the TIL? I think TILs are not so much genetically engineered like the CAR. So do you think the future is of the TILs? So should we shift towards the TILs rather than just crowding in the CAR-T research? For the solid tumor treatment.
Yeah, absolutely. Good point there. So actually, TIL is the first cell therapy that was done in the world, right, at NCI. Dr. Steve Rosenberg, he infused tumor-infiltrating lymphocyte from metastatic melanoma patient to, treat that kind of disease. However, TILs has its own complications. I agree with you that it's very lucrative because you don't have to engineer, and it comes from the tumor itself, so they are very likely to be reactive against the tumor, certainly. However, the complications exist. For example, it is not so easy to identify TILs from your tumors, right? First, you have to excise the tumors and then try to find the T cells or the T cell clone that are reactive to the tumors, right? It's already a challenging task. So, out of 100 patients you pick, maybe you will get maybe 20, 25 patients, that have the TILs that are reactive to the tumors. So the frequency is very less, right? Even though there are success stories, but the number of patients treated with this therapy compared to CAR T cell is much less due to this main reason. And another point is TILs, when they reach the tumor, they are already terminally differentiated. The T cells, they have their own lifespan. They start with something called naive cells, and when they encounter the antigen, then they start to differentiate into different T cell subsets. Some of them are memory stem cells, followed by central memory, effector memory, and EMRA population. And TILs, mostly, they are the terminally differentiated cells, meaning they do not live for a very long time. So you have a very narrow window. You should be able to harvest those TILs from the tumor and bring it in culture and then, expand them in large number and infuse back into the patient. So this may take a substantial amount of time. And by the time, TIL may give up in vitro. So that is another complication of TILs. But to answer your question in short, so I think TIL is a very lucrative approach, but we are not there yet. We need to find out, how we can identify these cells from PBMCs instead of, tumor itself. That would make our job much easier. And also, is there a method or a protocol to expand these cells in vitro in a very short duration of time so that they don't lose their viability and the potency? So these are, some of the key questions for the TIL field before, they come back to in a situation like the CAR Ts. So it's still, I think it will take some more time to reach that phase.
Let's move towards the solid tumors complexity. So how do you define the solid tumors complexity? How much complexity is, and what do you think are the challenges in the solid tumor treatment or the solid tumor based immunotherapy?
Okay, from the immunotherapy standpoint, right? So, do you mean to say from the CAR T standpoint or checkpoint, or
from the CAR T standpoint?
CAR T, okay, yeah, certainly. I think the whole world is moving towards this direction. The people are trying to find out how we can improve the efficacy of CAR T cells in the solid tumor context. But as you mentioned, there are a lot of complications. So, I would like to, put this complication in a different bullet points, various bullet points. The first one is the tumor heterogeneity, right? So the success of CAR T cell therapy so far is seen in heme malignancy. For example, the B cell malignancy. So they target something called CD19, which is a very clean antigen. It is not expressed by any other population except the B cells, right? So, when you use CAR T cells to kill this B cell cancer, so, it's very safe. They just kill the B cell and that's it. Now, coming back to the solid tumor, there is something called antigen heterogeneity. So meaning like it's not uniform. Even the whole mass of tumor may have the cells made of different expression level of your target antigen. Some cells may express at very high level, some cells at very low level. Some cells might be negative for this tumor antigen. So that is the first complication. The first complication is identification of the optimal antigen. And the second one is the complex microenvironment. Tumor microenvironment is very challenging in solid tumor. They have a hypoxic environment, limited supply of glucose, and then they have limited supply of other nutrients. And whatever is supplied, they are sucked up by the tumor cells itself, right? And also the penetration ability of the cells, it might be compromised in the tumor microenvironment. So there are a lot of complications. And due to all these reasons, your CAR T cells may get exhausted very early. They may get, compromised survival there, and even if they are able to survive, they may not function it well. So that's another complication. So to overcome this complication, a lot of work has been done by different principal investigators from industry and academic world. And some of the examples are development of cell platform that gives you better persisting ability in the solid tumor, expansion and persisting ability, and something called stemness feature. So people are working a lot in that field. So stemness feature is the ability of the T cells or CAR T cells, that helps them to stay for a very long time in the patient body, and also they help the CAR T cells to expand in the tumor microenvironment to achieve a better response towards the therapy. That's the second point, the complex microenvironment. And the third point is, all these sort of metabolic, suppressive environment and the limited, ability of the T cells to fight with the cancer from the metabolic standpoint. A lot of work has been done in that front as well. For example, Erica Pierce, who started the immunometabolism field, so she has done a tremendous amount of work, and also other peoples elsewhere, like Sukek and even Luca Gattimoni, they have done a bunch of seminal work in this field. So I would say that is another complexity. And the another complexity I would like to talk about is the pleiotropic nature of the cancer, right? They keep, the mutations, kind of how stable is the mutation, and how much downregulation of the target antigen is ongoing in the solid tumor. That's also another question. And also, kind of whether the CAR T cells you have generated can sufficiently recognize the tumor antigen expressed by the solid tumors or not. So these are the major complications. And apart from that, in the manufacturing scale also, there are a lot of questions, right? The traditional manufacturing process that we call, which is adopted by Gilead, Novartis, and BMS, that may not be a path forward to treat the solid tumors, because with the traditional manufacturing, CAR T manufacturing process, you generate cells that have lower potency to persist and expand. So that's why we have to change the manufacturing protocol as well, and also come up with a different armoring strategies. For example, you generated a T cells that have stemness feature, and you introduce the CAR molecule on the surface of the T cells. Now, in addition to that, you may want to arm your cells. We call it armoring of the CAR T cells. So we can do it in a different way, but for that, you need to understand what kind of tumor you are treating. For example, if you are treating hepatocellular carcinoma, which is a cancer of the liver, so in that case, your environment might be very hypoxic, very less oxygen may be available there. So in that case, you can design your CAR T cells in such a way that they have ability to overcome the hypoxic environment, and that can be done by silencing the hypoxic inducing factor inside the cells. Another example could be the TGF-beta, which is a negative regulator of CAR T cell function in tumor microenvironment. You can nullify, you can dampen those signaling by expressing dominant negative TGF-beta receptor. So there are a lot of examples like this. So, and also, we need to define our manufacturing process in order to achieve success in the solid tumor. So I think I spoke about a lot of things here, but just in summary, so solid tumor is very challenging. Everyone is on the same page regarding this statement, and some of the major problems are antigen heterogeneity, complex microenvironment, loss of antigen, and development of proper CAR T cells. The potent CAR T cells for the therapy through armoring mechanism. So these are kind of the field we should focus on in order to achieve success there, I think.
Like you told about the different problems, which is your the most favorite problem or the most challenging problem you think?
Well, all of this.
Yeah.
So all of this, I think like we have to tackle all of these problems. But to be honest, I have been in the CAR T cell for a very long time, but I did my postdoc research in something called T cell steadiness. Right? I had worked on a bunch of transcription factors and metabolic enzymes, that play a role in the T cell field. So I would say the first impactful work may come from this side, because if you are able to develop T cell that can sustain complex suppressive tumor microenvironment, your problem may be solved much quicker. Right? Even if you develop a very good armoring strategy, if your platform is not ready to fight in that environment, it may not be sufficient. So the first thing you should start from is to build a robust cell platform in order to treat more efficiently. So I would say development of a very powerful T cell platform for the expression of CAR and armoring molecules. That would be the first priority I would give to. So the most favorite problem for me in the solid tumor context is the complexity of the tumor microenvironment that forces cells to lose their surviving ability. And to overcome that, we should work on the development of more powerful and potent platform.
So when we talk about the platform, does it mean about the environment for the T cells? Like, do you think the combination immunotherapy could be an option?
Yes, certainly. Combination is something that is heavily worked on these days. For example, like if you infuse the CAR T cells and they get exhausted in complex tumor microenvironment, and people are starting to give checkpoint inhibitors in combination with CAR T cells, right? So certainly combination pathway, therapy pathway could be the another potential avenue in this field. And I do agree on that. And not only the checkpoint inhibitor, we may combine it with the radiotherapy and combine it with chemotherapy depending on indications and things like that. And sometimes, we briefly touch upon CAR macrophages, right?
Yeah.
So we may want to combine it with CAR T cell and CAR macrophages, so that we can send only the good macrophages to the tumor microenvironment, so that it generates a favorable environment for your CAR T cells to function in that complex tumor microenvironment. So yes, I think the combinatorial pathway could be the future in this field.
So the research is happening. It is not that research is not happening related to the hypoxia or the targeting the multiple antigens. I think this is happening, but still we are not getting the fruit of such a struggle. So why do you think this is happening? We are publishing the papers. There are a lot of papers. Sometimes the title looks very fancy, but still we in the market we don't find the CAR T therapy. So what do you think? What is lacking in this research?
Yeah, so first of all, you have to think from the another angle that this field is relatively new, right? So this field, the first CAR T cell was approved back in 2017, which was just 7 years ago, right? It's not that long ago, right? So that means we are learning, right? We are learning. So and it's not like we are not studying in the clinical setting. We are doing that as well, right? For example, I can take an example of many CAR T cells that have been infused into the patient with solid tumors and that have shown some promise. And just now AACR is done, right? AACR 2024, it was over just this weekend, and several promising data from the clinical studies were reported in that conference. So for example, I would like to talk about CAR T cell product called Claudin 18.2 from Astrazeneca. So this product was infused in patient with gastric cancer, and they have reported an objective response rate of about 50%. And remember, these CAR T cells were infused in a patient that were non-responsive towards all the existing therapy. Mm-hmm. So it's not like we are not achieving anything. We are starting to get some good results out there. For example, just in this AACR, another clinical data was presented from something called Cellular Therapeutics from China and US. So they have reported objective response rate of 60% in colorectal cancer, which is fantastic, right? It's a very aggressive disease, and still in that space with CAR T cells, they were able to see that kind of outcome. So I think the field is moving, and we are moving towards the solid tumor, and we are getting deeper understanding of the CAR itself, and also the platform and the tumor itself. And we are getting to, slowly starting to get some good data out there. And as you know, there are some proof of concept data available from glioblastoma, from renal cell carcinoma, from prostate cancer, right? So certainly there are some good stories that are coming out as well, but we have to wait and watch. I think the field is moving, and we will reach there.
Mm. What do you think about these kind of therapies? Can we ever get these kind of therapies very soon, or even if it is FDA approved, what do you think about the possibility of this kind of therapies in the countries like in Nepal or other countries?
That's a valid question because everyone should get access to the advanced therapy, right? So being a mankind, so we should be given an opportunity to receive this kind of therapy if required, right? However, I think it's very challenging in countries like Nepal. So there are a lot of kind of factors, that influence, or that negatively influence such kind of initiatives. So put the things together, I think the first point would be the awareness among the clinician and the public, right? So this is super new, super. Even if you talk with clinicians, they are new to this field. They will say, "What is this?" Some people will say, "Yeah, we have heard about it, and it's magical in heme malignancy." But how is it done, and how is it given? For what stage of kind of cancer patient it's given? At what stage it is infused? We get all this sort of naive question, and I think it's not anyone's fault. It's because kind of our education system is not there yet, and it's still in the exploratory phase, even in the developed world, right? So that is one of the factors which is kind of hindrance to bringing a new therapy to the country. Even if you bring, will it be viable? That's the question you will ask, being an innovator, right? And the second point is the regulatory, and which is a huge challenge, right? Because our regulatory authorities, first of all, we don't have a proper regulatory agency for biotherapeutics. We have something called, in Nepal, DDA, Department of Drug Administration, and they have a very minimal knowledge regarding this therapy. And the question here is how open they are to bring this therapy to the country. Are they ready? Right? That's another question. And the third thing is the facility, the infrastructure. I would not put infrastructure as a number one, kind of hindrance in bringing this therapy. I would put it at it as a third point, because if we have a group of right people, and if you have young people who are interested in learning this kind of a new field, then certainly we can teach and we can make it happen in our country. But the infrastructure is another hurdle. For example, even if you are able to produce these cells, do we have the clinics ready to facilitate the patient in taking these cells? That's a big question out there. For example, to take out the peripheral blood from your patient, we do something called apheresis. Can you imagine? There are hardly 2 or 3 centers in Nepal that do apheresis. Right? That's a very basic question. And the second one is, now, the side effects of CAR T cell, like cytokine release syndrome and neurotoxicity, we do see that, but it's not in that extent. In the beginning, we used to see a lot of CRS, a lot of neurotoxicity, but we don't see that much. But still, we have to be prepared for that, right? When we do the therapy for any patient. So you need a ICU and the tertiary care unit to take care of this patient, and do we have enough of this facility? So that's another question. And the other is the technical ability of the clinical manpower, right? You need nurses, you need the doctors, you need anesthesiologists who are trained in this. Are we prepared for that? I don't think so. So we have to provide training to these people as well. So I would say the major hurdle is the understanding of the field and the public awareness. And the second one is our regulatory spec, and the third is our technical hurdles. And the fourth one is obviously, whether we would be able to generate quality cells or not, right? But let's say people like yourself, who's very interested in biotechnology and bringing this field to the next level in the country, if you have that kind of motivation, then this fourth point can be easily solved, I think.
And what do you think about the cost burden for the patient? Because this is also the main problem.
Right. Right. So the cost is another factor. So in US, one shot of CD19 CAR T cell cost about $400,000, which is a lot of money. Even in US, not everyone can afford this, and it is not supported by the insurance system. And in country like Nepal, certainly, it's a challenge. But I think there are certain ways to, reduce the cost of this drug. The first one would be, we can use the rules or the law set by the World Trade Organization. Being a low middle income country, we have the ability to explore the patented drug in the Western world without any cost, right? That's a one point. And the second point is, you need to collaborate, certainly. Being from the developing world, you may not have access to all those resources, so you have to collaborate. And if you find a good partners who want to collaborate and who want to support us in getting, some of the very expensive reagents, then that might be really helpful, and that might help us to reduce the cost as well. And then the third point is, we can take the benefit of being in between India and China. These are the huge economies, and we know the reagents that are available, that are produced in the country, they are much less expensive compared to reagents from the US or from Germany or even from Italy, right? So we can exploit our geopolitical situation, and we can import the reagents from this country to substantially reduce the cost. And another one is the manpower, right? If we are able to train our own people, then I think we can produce these cells at much less cost, because we don't have to go through all those complex procedures like in the developing world. High insurance costs, high salary, and high cost of the reagent and the equipments that are required in the manufacturing facility. So with this point, I think like if we can implement, and if there is a support from the regulatory agency from the country, we can still do it in much less cost. For example, India has started to provide CAR T cells in 40 to 50 lakh Indian currency rupees, which is about 1/5 of what developed countries' patients are paying, right? Yeah. And again, they don't have the ability to use the IP product. They are using their own product because they are in the middle income country now, but still we have the ability to directly work on or produce the patented product from the developed world. So I think if we can exploit this point, we will be still kind of viable compared to the Indian counterparts. So certainly, I think it will be expensive, but we have to work on bringing the cost to about 1/10 or 1/12 of what developed country people are paying.
Yeah, I changed the topic completely. I think we were talking about the solid tumors, and I am now talking about this topic. I was in Nepal last time, and I think there was a conference organized by you and your team. So I want to know about your initiatives in Nepal and the CRM Nepal. What is it doing? And
sure. So what happened was, there is a beautiful story behind. There was COVID that hit the whole world back in 2020, right? Or late 2019. And everything was kind of not moving, everything was in its place. And back in 2022, I feel like, no, I should go back to Nepal and then just catch up with everyone. Before COVID had hit us, I used to visit the country at least once a year, and it had stopped for 3 years. And I wanted to really go back and get in touch with all the folks around and renew my, kind of networking. And then there was a conference organized by NAST. That was kind of, I think they do it once in 4 years, if I'm correct. And so I gave a talk there on, again, cell therapy. And I was sure that, it would be very complicated, the topic, but still, I gathered courage, and then I went there and said, like, okay, I want to talk on the cellular therapy. And then I spoke on that. And fortunately, the chair of the session was from the oncology field. And he invited me to give a talk in front of the doctors from Bir Hospital with his National Academy of Medical Sciences. And that's how it started. I started to get in touch with many oncologists and many clinicians, and we started to think about, hey, do you think it's feasible? Let's give a shot. Let's give a try, right? Because it's important, and looks like the world is trending towards this field. So we need to explore this as early as possible. So that was the conclusion. And that's how we started. We formed a team, and we started thinking about how we can bring it here. But as I mentioned before, that one of the major hurdles is the public awareness, right?
Yeah.
And also awareness among the clinicians about the field. So we thought of putting a conference there. And last year in April, we could do that, where some of the pioneer people were there speaking about the cell therapy. So that's kind of a story how it began, and we got a good response. And we thought of exploring the kind of a opportunity to bring this therapy in Nepal. So with this goal, we started an organization called Center for Regenerative Medicine Nepal, and currently we are 5 members, and we are working towards developing GMP facility. So the goal of this organization is not only to bring the cell therapy, but try to explore other modalities as well, including engagers and immuno-oncology modalities like checkpoint inhibitors. Yeah. So it's a very ambitious project, and we are really working hard to achieve that, and we'll see how far we can go. So at the moment, we are focused on developing a GMP grade facility, and we have achieved initial level of approval from the DDA. So we'll see when we're able to produce the cells in the GMP compliant facility. Yeah. So that's kind of overall story. And our goal is not only to work on the development of therapeutics, but also to train the new people and provide a conducive environment for the people who are interested in the field.
Yeah, I think that was a good platform. I also got a chance to know you more, know you better, and all the people from the field. So I appreciate that. So we are still sticking to the story and the talks related to Nepal. So talking about the cost, do you think the off-the-cell immunotherapy or the cellular immunotherapy could reduce the cost to some extent, or are the researchers still like doing this kind of research in the T cell or the CAR T cell?
Oh, absolutely, absolutely. So I think that's the path forward for the field, because cell therapy can be done in three different ways, right? Using three different platforms. First one is autologous, where the patient cells are taken out, they are engineered, and they are given back to the patient. That's autologous. Second one is allogenic, as you said, where instead of taking out cells from the patient, we take out from the healthy donors, and then genetically engineer them and infuse back into the patient. And third one is something called universal cells, effector cells, right? That is kind of an allogenic platform, but in allogenic, you have the limitation in the number of cells that you can produce. But in case of universal effector cells, you can generate trillions and trillions of cells and generate the same type of drug, exactly the same drug for many patients. And many pharma companies, many academic institutions and countries, they are working towards developing such kind of therapies. However, the immune system is not so simple, right? It has its ability to reject, which does not belong to the body, right? So that is the challenge in the allogenic field. And what people are doing is they are trying to use the CRISPR-mediated technology to knock out some of the molecules from the CAR T cells, from the donor T cells, that can play a role in rejection of these cells inside the body. So these molecules are like TCR molecules, major histocompatibility molecules, right? HLA-E and things like that. So yes, it is the future, we believe, and everyone is working towards that, including myself. I have one project within my team where we are working towards generating such kind of cells. And if it happens, certainly it will help to reduce the cost. How? Because with one product, with a one-time product, you can treat thousands of patients. So you don't have to remanufacture the cells again and again. So that will help to substantially reduce the cost. And the second one is it also helps for the patient to get access to the drugs much faster, right? Yeah. Now the turnover time for autologous therapy is 3 weeks. Means if you take out blood from a patient and generate CAR T cell, it will take 3 weeks for the patient to get back those cells for the therapy. So by that time, anything can happen to the patient because CAR T cells are normally given to terminally ill patient, right? Allogenic CAR T cells, if it gets materialized, which I believe will, then it will help to mitigate such kind of circumstances as well. That's the second point. And the third point is, it's off the shelf. You can find it anywhere. Yeah. You go to the pharmacy, you ask for a frozen vial, and you get it, and you keep it like a saline or kind of a blood bag, right? It's a very straightforward process. So yes, it will help to reduce the cost. It will help to provide an easy access of CAR T cells to all the people around the world. However, the complexity exists. The first thing is the allogenicity of these cells that might get rejected. And there are already a bunch of studies, clinical studies, that have been published in Lancet and New England Journal of Medicine that shows that allogenic CAR T cells have much less efficacy compared to the autologous CAR T cells. So one of the issues there is the allogenic CAR T cell capable enough to treat solid tumors? That's the question we have. Maybe it may not be an issue with the non-solid tumor, but it can be an issue with the solid tumor. So if we can overcome this point, then certainly it's the future of the cell therapy.
Yeah. So I think we are in the end. So maybe just a few questions I would like to ask. I think I should make a part 2 of this talk so that because I could not finish all the questions. So it is always inspiring to hear from you, your humbleness. I really appreciate that. And the way you make things clear, like in very easy language, and also the way you give the detailed explanation to everything I ask. So I really appreciate that. So the last question would be like, how was your journey? This is very important to know. And how was your journey from a graduate student to a leadership position in a big pharma like Gilead and Astrazeneca, and how did this magic happen? And what do you like to suggest to the young students, the young graduates?
Right.
Story.
Oh, yeah. Thank you, Sudarshan, again for this invitation. It is a fantastic conversation with you, and look forward to many more in the future. So coming back to your question, yes, it was not kind of a planned journey. I would say it happened on its own way. But there are certain factors that influenced me to take this path, right? So I would like to start from my, school time. So I studied in Pokhara. I did my IHC there, and I moved to India for my undergrad. I did undergrad in a place called Manipal, which is in the southern part of India, and I did lab sciences, right? And it was back in 2003 I started. And why I did that was because, at least, I thought like maybe I could come back and run a lab and, at least earn something for myself and my family, right? That was the goal. But as I moved to Manipal, I got exposure to, kind of a very good infrastructure and the faculty there. And, I got motivated to study further. And, I applied to many universities within India for my master program, like All India Institute of Medical Sciences, Banaras Hindu University, Indian Institute of Science, Indian Institute of Technology, Jawaharlal Nehru University, and, universities like all kind of the central universities of India. And fortunately, I got admission to some of them, and I picked Jawaharlal Nehru University to do Masters in Biotechnology. And initially, I was very hesitant because biotechnology was very new to our country. There was nothing, that was back in 2007, and still we don't have anything there. Yeah. That was kind of 18 years ago. And you can imagine how was the situation that time, right? And then I was like kind of very hesitant to join like biotechnology. Why? Can I do this, and should I do this? And I spoke with my faculties in Manipal, and they said, yeah, you have got JNU, so you should go there. It will open up a lot of avenues for you in the future. And I went there, and the initial days were very tough, very hard, because very good students were there from all over the India, and and I came from a little bit of diagnostic background, but there it was completely life sciences, genetics, immunology, molecular biology, and very complex science. And I was new to those, but still I gave my best, and I was able to do really well at JNU as well. And one of the factors that motivated me to do the higher study was my faculties there. I would like to still remember their name, Professor Rakesh Bhatnagar, who was the person who developed vaccine against anthrax back in 2001. So he was the dean of our school that time. And Professor Santosh Kar, who was a fantastic immunologist. I was kind of inspired by them, and I felt like, okay, I should do higher studies. So instead of, thinking about going back and starting to work, I thought of doing higher studies, and it was all because of the inspiration that I got from my faculty at JNU, Jawaharlal Nehru University. So I would like to give credit to my faculties for all, what I could do later on, whatever small thing I've done so far, it was all because of them. And so I applied for PhD program. Throughout the world, and I got admission to a bunch of them, and I picked Max Planck Institute and University of Freiburg in Germany because it was very well known for immunology, and it's still very well known. And I moved there, and I started working on transplantation immunology, neutrophils, acute graft versus host disease, bone marrow transplantation, and topics like that. And then again, it was a very fantastic opportunity to be able to do PhD in such a kind of a prestigious institute, and I could interact with many faculties like Mikhail Rath, who discovered the ITM motif, and Thomas Bohem is known as kind of father of developmental immunology, and also people like Wolfgang Schammel, who identified TCR clustering and all. So I feel very fortunate that I could work with them. And again, I would like to give credit to my JNU faculties. They pushed me to do immunology because they found out that I was good at immunology compared to other subjects, and I was fortunate enough to get opportunity to work in Germany as well. So when I was doing immunology, I was enjoying it a lot, and that time cell therapy was coming up. I could get opportunity to attend conferences and all, like Keystone and SITC and all, and I realized that there is practical implication, direct practical implication of cellular therapies or immunology, and I thought of doing postdoc, and I moved to National Cancer Institute for my postdoc, where I was initially postdoc, and afterwards I was a scientist there before moving to Gilead, followed by Astrazeneca. And when I was doing my postdoc, I was confused. I was thinking whether to go industry or academia, and I had got an offer from both the sides. So I had offered from Dartmouth College as tenure track faculty from University of Virginia, Charlottesville, and I also had offer from Gilead. I was a little bit confused where to go, but I thought, like, since the CAR T cell is very kind of practical, right? You can bring them to the clinic quite easily or quite quickly. So I thought of moving to industry, and I've been enjoying that so far. And it's a lot of differences compared to academic side, but I think for a good reason, because it makes you kind of focused, and it makes you understand the dynamics and also the politics to some extent, right? So I'm liking it. So coming back to your question, so how was the journey? The journey was very exciting, very thrilling, but challenging as well. So one of the challenges I faced was, I have traveled to 3 different continents in the name of study, and you have to adapt to the new culture, new languages, new food and all, right? And I was away from home, and I left my home when I was 18 years old, and it has been very challenging. That's the first thing. And the second thing is to kind of, understand the mental side of different geographical region, right? That was quite challenging. First, I was in South India and North India, completely different, and then in Europe, in America, right? Europe, it's very straightforward. You say no to no, but in US, no to no is no does not happen, right? So that kind of adaptation always happened during this course. Nevertheless, I feel very satisfied when I look back. So I think the most important thing, to advance further in life or in career growth is perseverance. You need to keep doing what you're doing, and you need to enjoy the things you do. You don't need to be a topper. You don't need to be a kind of a board first or things like that. It's good if you do really well in studies, but more than that, I would give preference to perseverance and continuity. And I think if you do that, you will reach where you want to reach. And the journey is still ongoing. I'm still a learner. I'm still a student, and I still try to attend conferences, talks, lectures given by the people around, and I enjoy speaking to all my team members in my current work, starting from undergrad to kind of postdocs to scientists, senior scientists, and I keep learning from them as well. It's you can learn not only from the people above you, but also from the people who are fresh to the field, right? I'm enjoying that process, and the overall goal should be to continue doing what you do and love the work you do, or find the work that you love and love the work you do, and also learn from everyone around you. And I think that's the most.
Let's pause this for a moment. And you said that you are a learner, you are a student. Suppose if you are given an opportunity to be a student again, a graduate student again, so what kind of research would you do, and what kind of skills would you learn? I think. I think that's a good question, because when I look back at my PhD or my master's program, I feel like I should have done something in a different way, right? So one of the things, learning part is okay, science part is okay, education part is okay, practical part is okay, research part is okay, but more than that, I would say the soft skill is also important, right? You need to know how to deliver your work or science, to the public, how to make them understand. That's one point. And the second point is you need to know the people. You need to know compassion. It's very important, right? Initially, I used to think, learning science is everything. No, that's not true. It's very important. It helps you to grow further in your scientific arena. But more than that, you need to know the people. You need to understand the people. You should build a habit of listening, right? You should build a habit to kind of, hear the things with compassion. And I think you should complain less. Right? So complaining does not help. Right? You should take every failure as a learning, not as a failure. I've failed many times, a bunch of times. Initially, I used to take it personally, but that's not the way. I think you should take it as a learning and move on. These are very important things in life, to not only to be successful, but to keep yourself, sane, and keep yourself mentally peaceful, and also, kind of to make your emotional health in good shape. So these are very important things, and you should start learning this thing from your PhD time, which is the crucial time where you start to interact with people for your research, right? You can think about other stuff as well during that time, and trust me, it will help you a lot down the line. Thank you so much, sir. Thank you so much.
No problem. Thank you so much, Darshan, for this wonderful chat, and look forward to staying in touch.